My name is Tatiana Gonzalez. I've been interning with the Gregory Lab for the past 2 months, and focused my research on the effects of hormonal therapy on the single cellular level. And before I go into that, I'd like to talk a bit about HuBMAP and our contributions. So HuBMAP, the overall goal is to basically study human tissues down to the single cellular level and to be able to create a three-dimensional map of those cells. I find this really interesting because I think it will give a lot more insight on how cells work together, and as well as to understand where they are actually located spatially, and the genes that are involved with each cell. Another thing I find really special about this project is that we're just such a diverse group of organisms. So we have to make sure we study both sexes. We need a variety of ages, a variety of ethnicities, because all of our bodies are different. And that leads me to what our lab was focused on, which is the female reproductive tract. As Stephanie mentioned, we'll be studying fallopian tubes, ovary, uterus, and the cervix. The way we study this is we have organ donors who will donate their organs to us, and some of the requirements that we have are that the organs come from someone who is premenopausal, and that all of the organs are from one donor, just to provide consistency throughout our research. Something interesting that happened, though, is one of our organ donors is actually undergoing a gender reassignment. So they're being hormonally treated through testosterone therapy. And I wanted to see what those effects had on the cervix. So basically, testosterone and hormonal therapy, the goal of this is to bring estrogen levels down and testosterone levels up. And I think it's important to talk about what average testosterone levels are for a female, male, and then female transitioning to male. So for a female, it's 15 to 70 nanograms per deciliter of blood. And for male, testosterone levels range from 280 to 1,100 nanograms per deciliter. And then from female to male, the testosterone goal is 400 to 700. So you can see it's a big jump from 15 to 70 and going to 400 to 700, which led me to my research question: how does this affect the cervix on the single cellular level? In order to conduct my research, I had to set up a few goals. First was to determine all the cell types present in the cervix. Next was to make comparative observations between cell types in a hormonally treated cervix versus a non-hormonally treated cervix. And finally, it's to research why certain genes are expressed more than others between the two, and what that means in terms of hormonal therapy. So here we have our methods. First, this is a very broad method list, just for the sake of time. But first, we have tissue dissociation, where you're slicing the organs into very small sections for higher resolution to see more in detail what cell types are there. Next, we have nuclear isolation, which is the actual breaking open of the cell in preparation for gene expression analysis using 10x genomics. And that's where I come in here at step 3. And for our final step, it's spatial transcriptomics using FISH. Our lab has not gotten to that step yet, but that is kind of our end goal. Okay, so here we have a HuBMAP of what seems like a typical cervix. So each dot represents a single cell, and they're clustered together in terms of similarity to each other. And what I meant by a typical cervix is one that has not undergone testosterone treatment. And this one actually has undergone testosterone treatment, and it is this little cluster here in purple. We call it cluster 13. And what's special about this is that this cluster here is unique to cervix that has undergone testosterone treatment. So one that would not have this cluster here at all. So this is kind of like the focal point of my research. As you can see here, this cluster is isolated. Over to the right, we can see it's cluster 13, that's what we're calling it, and it only has 120 cell types in that cluster, which is the least amount in comparison to the rest of the clusters. What I really want to focus on is this area down here, and this is our gene expression. So the reason I included this picture is because for comparative purposes, if we see here at cluster 13, we can see how upregulated these 3 genes are. But when you go across and look at the other clusters, they're very downregulated in comparison. So we'll look at the first one, which is MIR31 or MIR31. In cluster 13, this gene is upregulated 9.38 times. And given the p-value, it is highly significant. So when you look over here, these are all in the negatives, meaning they are very downregulated and not being expressed in these cell types. So I found it very interesting that the genes being expressed in cluster 13 are very, very different from genes being expressed in the rest of the clusters of the cervix. And I will go over 3 of them for the sake of time. Okay, so what gene expression tells us about effects of hormonal treatment in the cervix? So first, we are starting off with MIR31. This was the most upregulated gene, and its expression is often linked to cervical carcinoma as well as colorectal cancer. And it is also linked to Duchenne muscular dystrophy, which is basically the degradation of muscle and the weakening of a muscle, which is quite interesting to me because the cervix is a muscle, and during menstruation or during giving birth, you need contraction there. So the fact that undergoing this testosterone treatment is kind of hindering that contraction is quite interesting to me. Also, it has oncogenic properties, which means it's tumor inducing, and it also has immunoevasive properties, which means it suppresses your immune system. Next, we have MUC16, which is the second most upregulated gene. So this too is overexpressed in multiple cancers. It causes tumorigenicity or causing cells to create benign and malignant tumors. And it also increases rate of metastasis in several malignant growths. And finally, the third most upregulated gene, REX, it regulates erythroid cell expansion, and erythroid cells are basically red blood cells, and it promotes formation of hemoglobalizing erythroblasts. And it is, something really interesting about it is that in comparison to the organs in the female reproductive tract, it is most expressed in the cervix. So basically, the importance of this study and what this tells us is that based off that cluster 13, there's certain genes that are being expressed that normally or typically shouldn't be there. And as we've seen with the top 3 genes that are most expressed, some of them can be quite detrimental to health and can just be concerning in general. So this information could provide us with insight on how to prevent risk of overall health for anyone undergoing hormonal therapy. And I think to further this research, it would be really interesting to see what genes are expressed in other organs other than the cervix, and see what those effects may have on anyone undergoing that treatment. So yes, I would love to give a big special thank you to the NIH and HuBMAP for my opportunity here, to the O'Neill Lab for getting us those samples, and a special thanks to Dr. Brian Gregory and Rongwu for taking me under their wing, involving me in their research, and just teaching me so much this summer. And yeah, that's about it.